Hello, everyone, and welcome to our educational webinar titled Exploring the Impact of SARS-CoV-2 Infection on the Central Nervous System. My name is Amanda Vandenhoek, and I'm a Senior Product Manager here at STEMCELL Technologies, and I'm going to be your moderator for today's webinar. It's my pleasure to introduce you today to our speaker, Eric Song. Eric earned his Bachelor of Science from the University of Southern California and completed his master's degree at Johns Hopkins University, where he worked on developing strategies to help treat chronic diseases, such as pediatric brain tumors. Since 2016, he has been studying immunological approaches as therapeutic strategies against brain tumors in the lab of Dr. Akiko Iwasaki. Following the COVID-19 pandemic, Eric has now been researching neurological impacts of SARS-CoV-2 on the central nervous system, and we'll be talking today about his latest findings on the neuro-invasive potential and neuroinflammatory implications of this virus in both mouse and human brain organoid models. If you'd like more information about Eric's scientific background, you can read about it in the link that is sent around in the chat. Let me now hand you over to Eric to begin today's presentation.
Thank you, Amanda and Kimia, for having me give this talk on learning about the impact of SARS-CoV-2 infection on the central nervous system. So my name is Eric. Once again, I'm from Akiko Iwasaki's lab, and you can find our Twitter accounts here for any more information. So as everyone knows, this COVID-19 and the virus that causes COVID-19, SARS-CoV-2, has caused a global pandemic affecting millions of lives all across the world. I'm not going to go into too much detail about the actual virus and the disease, since most of you guys might be more of an expert than me. But it's primarily a respiratory disease causing respiratory dysfunction and ARDS-like disease. It has many similar characteristics as another coronavirus that we experienced in the early 2000s, SARS-CoV-1. And similar to SARS-CoV-1, it affects multiple organs, causing multi-organ dysfunction. But it's still very debated whether it's a primary invasion by the virus into these organs, or it's a secondary outcome, such as cytokine storms or other immunological dysfunction that's causing a global disease. Unlike SARS-CoV-1, the main difference is how widely affected this disease has affected patients all over the world. And because of this, there's been a lot of data that was not available actually for SARS-CoV-1, and a lot more different variety of symptoms that we're noticing in this case. And one of these, of course, what we're going to be talking about mainly today is the neurological symptoms associated with SARS-CoV-2. So since the outbreak, there have been many different symptoms associated with neurological central nervous system dysfunction. And like I said, like the other organs, it's unknown whether this is due to a primary invasion of the virus into the central nervous system or due to like a secondary effect. The symptoms are very variable. So people have noted anywhere from like loss of smelling, loss of taste, to more serious symptoms like viral encephalitis-like symptoms, strokes, and other things. And many groups have really taken on the effort to understand what the possible implications are. And like I said, it ranges from neurologic to neuropsychiatric complications. And many prospective studies have shown that up to 30 to 60% of patients with COVID-19 demonstrate some level of neurological manifestations. So, but there's still a lot of ongoing questions regarding SARS-CoV-2 in the central nervous system. Autopsy studies here have shown that you can find SARS-CoV-2 transcripts at high levels in the lung and pharynx of patients, postmortem patients. And you can find RNA levels of SARS-CoV-2 in the brain as high as other commonly kind of thought of organs, such as like heart, kidney, liver. But it's still really unknown that whether this is a primary invasion, or you're just picking up, viral RNA in circulation or something like that. However, there's enough evidence to make us believe that SARS-CoV-2 can infect the brain. So during the initial SARS outbreak, people have looked at the possibility of SARS and its neuro-invasive properties. So for example, here taking a patient who died from SARS infection, they took the lung and the brain, and they were able to find PCR from the homogenates of these organs of SARS-1 transcripts. Taking the brain homogenate and co-culturing it with cells in vitro, they were able to see that the homogenates had viral particles that were infectious, as evident by the EM images there. And other groups similarly showed that in postmortem sectioning of brains from the SARS patients showed positive staining for the virus, indicating SARS can actually have a neuroinvasive property. Similarly, SARS-CoV-2 also utilizes similar receptors and have many of the same properties like we discussed. So it's not too far-fetched that you can imagine SARS-CoV-2 could also have a neuroinvasive potential. But this was all done kind of in the early 2000s, and since then we've had major advances in technologies and the way we view neuroimmunology. So we decided to apply some of the newer techniques and newer ideas in order to answer questions regarding SARS-CoV-2 neuroinvasive properties and neuroinflammatory effects. So today I'm going to be touching up on three main topics and trying to answer these three questions. First is, how can we study SARS-CoV-2 in a laboratory setting? Second is, what is the neuroinvasive potential of SARS-CoV-2? And the third is, what is the neuroinflammatory consequence of SARS-CoV-2? And going first with the first one, how can we study SARS-CoV-2 in a laboratory setting? In addition to having to gown up in like BSL-3 facility clothes and going through all the hazard things for working with SARS-CoV-2, there's some additional kind of challenges in trying to actually study the virus using different model systems. The main issue for us was that since we're very keen on using mice models, because mice provide us with a wide variety of genetic backgrounds and such to be able to kind of look at the basic biology of different viruses, SARS-CoV-2 cannot infect wild-type mice. This is because, similar to SARS-CoV-1, it requires a human ACE2 receptor for actual infection. In order to get around this, the most famous mouse, I would say, is the transgenic K18 human ACE2 mice, where human ACE2 is overexpressed under a keratin 18 promoter to be able to make the mice susceptible to the virus. And this was made initially for the initial SARS outbreak. But since then, there have been, during this outbreak, there has been several other versions of a transgenic human ACE2 mice that have been developed. However, using a transgenic mice requires long breeding time, and to be able to study in different backgrounds of different like immune knockout or immune sensor knockouts, it requires long breeding time to be able to get like homozygous mice. And for example, like it would take months to be able to get a K18 human ACE2 knockout plus IFNAR knockout mice. Also, when we started the project, it was back ordered from JAX, and even now, you only get limited amounts from JAX to be able to study with this mice. So we decided to take on an approach done by Dr. Perlman for MERS here, in using a viral vector to be able to express the receptor of the virus for the virus infection to occur. So our group and two other groups, including Dr. Perlman's group, use a viral vector to be able to overexpress the human ACE2 in mice to make them susceptible to SARS-CoV-2. So in our lab, we decided to use an adeno-associated virus because it's known to be a little bit less immunogenic than adenoviruses. And by using it, by using a little less immunogenic virus, we hope that it wouldn't interfere with our SARS-CoV-2 infection. So we transduced the mice with a human ACE2 AAV, and then 2 weeks later, we would challenge the mice with SARS-CoV-2. And by overexpressing human ACE2, we were able to show that mice had observable titers of SARS-CoV-2 in the lung done by plaque assay. These mice also demonstrated innate immune cell infiltration and a more activated state of the innate immune cells. Carrying this out to day 7, day 14, we checked for antibodies against SARS-CoV-2 in the serum of these mice and saw high amounts developing. So this indicates that by overexpressing human ACE2 using a viral delivery system, we're able to make the mice susceptible to SARS-CoV-2 and the infection level, and see innate and adaptive immune system engage the virus over time. So to look at what happens when the virus infects the tissue, we did bulk RNA sequencing to check for what kind of genes were upregulating in this mice. And we saw that the differentially expressed genes were mostly ISGs, or interferon stimulated genes, similar to gene signatures that were present in patient postmortem samples. And we noted that most of the ISGs overlaid with the type 1 interferon signature. So then to see how the importance of type 1 signaling, we looked at IFNAR knockout and IRF37 knockout mice by transducing them with human ACE2, and this was one of the advantages of using a adeno-associated virus system, because we were able to test this kind of simultaneously with the wild type mice without a long lead time. And we saw that the ISGs were completely abrogated in these mice, and they saw less immune cell infiltration. So you can read more about that project in the mouse model of SARS-CoV-2 reveals inflammatory role of type 1 interferon signaling. This was done with a postdoc in our lab, Ben, and I led the project. So I really wanted to kind of prime the whole webinar with that, because I wanted to show how these 3 kind of independent questions all had aspects of experimentation and stuff that interplayed with each other that really kind of synergized and made each story much better. So thinking about the central nervous system component now, I'm going to go into what the neuro-invasive potential of SARS-CoV-2 is. And this started from this K18 mice that I briefly discussed. And we were able to obtain a few early mice to study the effect of SARS-CoV-2 in these mice. And interestingly, when we challenged them intranasally, we saw decreasing titers over time in the lung, but in the brain, we saw significantly increased titers over time. This is actually very similar to what was observed in SARS-CoV-1 done by Dr. Perlman in the early 2000s. And this indicates that the brain can harbor SARS-CoV-2 and has the machinery required to allow for significant proliferation. Now, when we look at the staining of the brain of these mice, we can see in red significantly stronger signals through day 4 and day 7. And then we see microglia that go from like ramified states into more amoeboid shapes overlapping with the virus, indicating there's some sort of immune response going on. So this is great, and it's interesting that SARS-CoV-2 has this brain potential, but using K18 mice, you can't dissect out the role of SARS-CoV-2 in the lung versus the brain at any given time in a mouse. So this is where we thought the AAV model would be perfect to be able to compartmentalize the infection into two different organ systems. So what we did was we would introduce the AAV human ACE2 into only the brain by injecting into the CSF of the mice. So then you would make the brain susceptible to SARS-CoV-2. Or you can introduce it into the trachea to make the lung susceptible to the SARS-CoV-2. And by doing this, we can challenge specific organ systems and see what the outcome of the disease would be. Surprisingly, what we noticed was that in the lung-only infection shown here, the mice didn't lose weight and the mice didn't succumb to any viral burden. However, when the human ACE2 was expressed in the brain and virus was introduced into the brain, we saw significant weight loss up to like 70%, and then all the mice had to be euthanized because of severe encephalitis-like symptoms. Even at doses low, considered here is about 100-fold lower than the intratracheal doses, the virus was showed high mortality in the mice. So it was very interesting to see the neuro-invasive properties of SARS-CoV-2 in mice, but we wanted to better see if it's also true for humans. And we can't really just go around injecting patients, but since the early 2000s, a technology that has really blossomed is the use of human brain organoids to be able to model different human brain diseases. So we decided to model SARS-CoV-2 neuroinvasion using human organoids, and to make human brain organoids, interestingly, we use a lot of stuff from stem cell tech, and I don't think that was planned in any way, but that's just how it ended up being. So my co-first author, Cha Zhang, and my classmate, he grew these organoids for a very long time for his other project, and we decided to use some of it to see if it can be infected by SARS-CoV-2. So our group was not the first to actually show this. There was two other bioRxiv papers that came out during the time we were working on this project, and we saw SARS-CoV-2 can infect the organoids in very widely. So here you can see in both SOX2 indicating more neuroprogenitor-like cells overlay with the red of SARS-CoV-2, and also MAP2, which is more differentiated neurons seeing infection. With the help of a collaborator, we want to see on a more granular level where the infection was occurring. So we performed EM, and we saw very hot spots of viral replication in certain neurons indicated here. And then we were also able to see that interestingly, the viral particles were budding off of a lot of ER Golgi-like structures, indicating that the virus was able to use the host machinery to continue to replicate itself during this time. And importantly, we wanted to see if SARS-CoV-2 can actually cause neuronal death. So we, with the organoids, we did tunnel staining to be able to visualize here in green which cells were going through some sort of cell death. And strikingly, we saw a lot more cell death occurring in organoids that were infected with SARS-CoV-2, as you can see here. But interestingly, there was almost no overlap between tunnel positive cells and SARS-CoV-2 positive cells. When we quantify it, most of the cells that were going through cell death were actually coronavirus negative. And to just kind of make sure that this wasn't like an artifact, we also looked at, is this only happening in regions with SARS-CoV-2? And tunnel positive cells were closer in proximity. So this is tunnel positive cells by percentage in regions where there was higher SARS-CoV-2 density, indicated by the x-axis, showing that it definitely seemed to be SARS-CoV-2 specific, but not in SARS-CoV-2 infected cells. So in order to see what was going on a cellular level, we took the approach to sequence these human brain organoids. By using single cell sequencing of the infected human brain organoids, we were able to identify broadly different types of mature and more progenitor-like cells in these organoids. And counting the transcripts of SARS-CoV-2, we saw that after 96 hours post-infection, we saw wide distribution of SARS-CoV-2 and almost no specificity in what kind of cells it was able to infect. To see what the transcriptional changes were, we first started with a broad analysis of what the transcriptional programming was in SARS-CoV-2. We decided to compare it to a more well-known neurotropic virus, Zika virus. So we looked at genes that were upregulated in Zika virus, and then overlaid it also with genes that were upregulated in the SARS-CoV-2 infected organoids. And to our surprise, we saw basically no overlap between the two gene programs. Taking the two gene programs and putting it into Go ontology or gene ontology programs, and then we noticed that both infections had processes that have to do with infection, right? Like response to stress, response to biotic stimulus, and some sort of foreign invasion was occurring. However, while Zika was going through primarily type 1 interferon responses, interferon gamma responses, and other like cytokine mediated signaling pathways that we know well about, SARS-CoV-2 was not enriched for those pathways at all. Rather, they were going through pathways related to cell division, organelle fission, cell cycle, metabolic processes, and protein targeting to the ER. So this was very kind of like surprising to us. It is known that neurons have a different capacity to invoke immune responses compared to other more like other stromal or immune cells. But because Zika was so immunogenic in neurons, it elucidated like this possibility of the neurons being able to invoke an immune response. But in the SARS-CoV-2 case, this was definitely not the case. So we looked at within an infected organoid, we decided to check the transcription changes in the infected cells, so those cells that had SARS-CoV-2 transcript reads versus cells that were just bystander or neighboring cells to these infected cells. In looking at SARS-CoV-2 positive cells, we saw what we expected, like viral transcription, nuclear transcribed mRNA catabolic processes. But interestingly, the top pathways had to do with like aerobic electron transport chain, cytochrome C to oxygen, and a lot of like metabolically active genes. In addition, this pathway response to hypoxia came out. When we looked at the neighboring cells, they were going through regulation of cell death, which we noticed in our staining, but additionally, they were going through kind of alternative metabolic processes such as secondary alcohol, mitochondrial protein catabolic processes, and cholesterol biosynthetic processes. And they were going through cellular response to hypoxia and response to decreased oxygen levels, which is a counterpart to what was happening in the SARS-CoV-2 positive cells. To see if this hypoxia was actually occurring in the organoids, what we did was we looked at mock infected and SARS-CoV-2 infected organoids and stained for HIF1 alpha. Indeed, we noticed that the infected cells had significantly higher HIF1 alpha positive cells in the organoids, indicating that the sequencing we did was kind of true. So what we imagine is happening in this case is that SARS-CoV-2 as a virus cannot invoke a sufficient immune response or cell death pathway in the neurons that it's infecting. Rather, it's using the neurons as like a vesicle to replicate itself, right? And during these kind of like highly replicative settings, the neuron requires a lot more resources from its surroundings, such as oxygen or glucose. And by doing that, it's sequestering it away from its neighboring cells, causing the neighboring cells to go through hypoxia-like gene programming, making them more susceptible to cell death. So we kind of touched upon this, like what happens when the neurons get infected by SARS-CoV-2, but there's this big debate about how does a neuron get infected, and because most of the cells that are susceptible to SARS-CoV-2 is due to this famous receptor ACE2. People have been doing many transcription studies, or looking at transcription data to see if ACE2 is highly expressed in neurons, and it's not. Especially on the mRNA level, people have been debating whether ACE2 is actually expressed in neuronal cells. In our data set, we also noticed that ACE2 mRNA level was very, very low, and it didn't correlate at all with which cells were getting infected in our single-cell RNA-seq. However, when we stained the organoids with ACE2 antibody to look at the protein level, we saw high levels of ACE2 in both the neural tubes by these like round structures, and in the mature neuron areas, indicating that there is ACE2 expression in neuronal cells, and also that this might be why the cells are susceptible to a SARS-CoV-2 infection. To check if it was using ACE2 to infect, we first treated organoids with either isotype or ACE2 antibodies to block ACE2 receptors, and then infected them with SARS-CoV-2. And you can see here the isotype treated organoids infected well, while the ACE2 treated, anti-ACE2 treated organoids were no longer infected by SARS-CoV-2, indicating that SARS-CoV-2 uses the ACE2 receptor even in cells, neuronal cells. So you can read more about this one on the neuro-invasive potential of SARS-CoV-2 revealed in the human brain organoid model. And we really wanted to kind of look at what were the requirements for SARS-CoV-2 infection in the mouse and a human brain organoid model, and look at what happens to the neurons when it infects. However, the organoid system is, is great because it has so many diverse cell types, but it's missing a large compartment of the cellular types that we find in humans or in an organism. And this is my primary interest in understanding the neuroinflammatory responses during different neurologic diseases. So we wanted to look at what the neuroinflammatory consequence of SARS-CoV-2 may be, and really focusing on how it may differ from systemic responses that occur when there's a SARS-CoV-2 infection. So to study the central nervous system specific immune response, we are very fortunate to receive lumbar puncture samples, CSF samples from COVID-19 patients and healthy volunteer patients. So what we did first was we would centrifuge the cells, and we would use the cells for single cell sequencing, and we would use a CSF supernatant for various assays. Oh, and what we noticed was that when we first did a cytokine panel to look at different cytokines that might be overexpressed, we thought we might find that the CSF might mirror what's going on in the systemic circulation. People talk about different cytokine profiles, and interestingly to us, this was not the case. And in fact, we found cytokines like IL-1 beta and IL-12 that were specifically upregulated in COVID-19 patients' CSF, but not upregulated, maybe even trending downwards in plasma samples of the same patient. So we wanted to kind of figure out what was going on in the two compartments. So we performed single-cell RNA sequencing and matching CSF and PBMC of the patients. We were able to identify the general cell types like CD4, CD8 B cells, CD4 and CD8 T cells and B cells, along with different innate immune cell innate immune cells. Since we saw IL-1 beta and IL-12 upregulated, we wanted to first focus on the innate immune cells. We can see that IL-12 has detectable reads in these myeloid 1, NK cell, and dendritic cell-like cells. And we wanted to look at what kind of changes were occurring in the CSF compared to the rest of the compartment. So in order to do this, I'm going to kind of take you through this graph. We decided to visualize it in it's kind of a bar graph version of a Venn diagram. So what you can see is the first column indicates genes that are specifically upregulated in the CSF of COVID-19 patients versus control patients in the different cell types. Then the second column will show genes that are specifically upregulated in the PBMC of COVID patients versus control patients in different subtypes. And then you can see later shared genes between CSF and PBMC, both upregulated, both downregulated. And then you can also see unique and shared gene profiles for a multitude of combinations. And you can see here, while CD16 and CD14 monocytes are going through transcriptional changes that indicate a specific downregulation of genes in the PBMC compartment only, cells like dendritic cells, myeloid 1, and NK cells are going through transcription profiles that seem to be very specific to the CSF or the PBMC in the case of NK cells, indicating even though it's a single infection occurring in the body, you can have two different immune responses occurring simultaneously in a central nervous system compartment versus the rest of the body. To look at what changes are occurring in the cells that were kind of highly expressing IL-12, we looked at the COVID-19 CSF dendritic cells and saw that these dendritic cells were expressing more type 1 interferon, consistent with the fact that we're finding more IL-1 and IL-12 in the CSF. The NK cells were also more activated compared to the PBMC compartment. You can see gene set enrichment of like pathways related to NK cell mediated cytotoxicity and chemokine signaling pathways in the CSF while it's not enriched in the PBMC. To kind of look at whether this innate immune cell change was affecting the global immune population of the CSF compartment, we decided to use analysis called cell phone DB. So cell phone DB utilizes known and predicted ligand receptor interactions and to use single cell RNA sequencing data to say, oh, these genes are upregulated, so there may be more interactions occurring. And you can imagine if more interactions are occurring or immune landscape that requires more immune cell interaction will provide an enrichment of certain interactions. So in our case, what we did was we looked at the predicted interactions in COVID patients and control patients in the CSF and counted as if there was or looked at whether there was increased interactions in COVID patients to indicate that there may be an immune response occurring in the CSF of the COVID-19 patients. And we saw that NK cells, dendritic cells, and the monocytes were interacting more with the CD8 and a little bit less with the CD4 compartment and almost none with the B cell plasma compartment, indicating that the COVID-19 patients had a general inflammatory milieu and in the CSF, and there was increased interactions. Because it highlighted the T cells, we we want to dive a little bit more deeper into what was occurring in the T cell compartment in the CSF. And when we subsetted the T cells from our previous data set and looked at the transcriptional changes that were occurring, it was very heavily skewed towards unique immune or unique changes, transcriptional programming changes occurring in the CSF compared to the PBMC, as you can see here. To look at what kind of changes were occurring, I can show you some of the data on, first we looked at CD4s, and then we looked at what genes were differentially expressed in the CSF of COVID-19 patients versus the PBMC in the COVID-19 patients. And then, like, for example, in the Th1 compartment, we saw chemokine receptors and no markers indicating a more effector-like state of Th1s. In the Th2, we also saw increase in ISGs and other integrins and chemokine receptors, indicating they were also more active in the CSF compared to the PBMC. And interestingly, when we took the shared gene profiles, we thought this can tell us a little bit more about what was happening in the CSF as a compartment compared to the PBMC specifically. And putting this through gene ontology, we saw enrichment of interleukin-12 and IL-1 mediated signaling pathways, which is consistent with the fact that we were finding these cytokines upregulated by ELISA in our first experiment. So there's a lot more other transcriptional changes that occur, but we also wanted to look at whether the because we did a 5' 10x sequencing, we were able to see the repertoire of the T cells to see if there's any clonal changes in the CSF compared to the PBMC. So similar to the cytokine theory, we initially thought we might see a mirroring of the CSF TCR compartment to the systemic circulation. So what that would mean is the top clones that we find in a certain compartment may be very similar to the top clones found in the other compartment. But this is not the case. And showing here, the first UMAP plot shows that the top clone in the CSF was the third top clone in PBMC. So that would be consistent with the idea that the two compartments mirror each other. However, we were able to find clones that were, the 13th largest in the CSF, but it was the 4th largest in the PBMC, and then vice versa, where it was the 4th largest clone in the CSF, but the 21st largest clone in the PBMC, indicating that even against a possible similar pathogen, you have like kind of preferential, pockets of T cells that are expanding in one compartment compared to the other. And you can see even here, there was a clone number 5, the 5th largest in the CSF compartment, but it was not found at all in the PBMC. And when we look at CD4 expansion, the largest clone found in each compartment, so here we indicated the shared clones that you can find in both compartments, like shown in 1, 2, and 3, as a mobile clone, because it can be in multiple compartments, versus like a clone that's only found in one compartment as a resident clone. And you can see only in the COVID-19 CSF, you find the statistically enriched CD4 clone size, indicating that you can have a local immune response that differs from the rest of the body and is not shared with the systemic circulation. So I'm gonna switch gears for a little bit and kind of show you the B cell side to this story, because it's very mirrors what we found in the T cell compartment. The B cells were particularly interesting because it was like the only cell type that was increased in frequency in the CSF compared to the COVID-19 or in the COVID-19 patients compared to the control patients. And you can see we had one patient with an extremely large number of plasma cells found in the CSF. So when we looked at the clones found in that patient with the expansion, we saw that clone 1 in the CSF accounted for almost 10% of the BCR clones in the CSF, while the top clone in the PBMC of the same patient accounted for about 25% of the BCR. Interestingly, the top clones that were found in each compartment were just completely different sequences, indicating that the two, again, do not share or do not mirror the immune responses that are going on at any given time. But importantly, in both cases, in all the patients, we were able to find high amounts of IgG against the spike protein of SARS-CoV-2 in the plasma, and interestingly, also in the CSF, except for one patient here, indicating that we might be having SARS-CoV-2 specific immune responses in both compartments, but the way our body is handling it might be slightly different in terms of what kind of immune cells are targeted to each compartment. To look at how the antibodies, if the antibodies were able to provide protection against SARS-CoV-2, we took the antibodies and used our organoid system to check its neutralization activity. We were able to find high amounts of SARS-CoV-2 antibody in the COVID-19 patient CSF, shown by the dilution factor here. And similar to the anti-ACE2, by pre-treating the virus with the SARS-CoV-2 or the COVID-19 patient CSF, we were able to almost completely neutralize the SARS-CoV-2 infection activity in the human brain organoid system. As of this recording, we're showing that, or this project was not actually published yet, but it'll be up and for you guys to kind of dive into the data a little bit more. But we really focus on what the neuroinflammatory consequence of SARS-CoV-2 is, and for the first time showed that you can have a sense, even within a one body or even in one organism, you can have two different immune responses that are occurring in the central nervous system versus the systemic circulation. And I think like combine these stories tell the importance of trying to understand this virus in a more global level. Like we're not just treating SARS-CoV-2, but we have to start thinking about how SARS-CoV-2 affects different compartments of the body differently, and come up with plans in understanding how we can treat each compartment uniquely, because the kind of the pathology or the immune response that's occurring in each compartment can be severely different. So that's it. This was a huge team effort. There were so many people involved with making these projects happen. Of course, Akiko allowing me to do these great projects and very interesting and important projects in the lab. Ben, who's a postdoc in our lab, we worked on the animal model together. Cha, my classmate from Kaya's lab, we did the organoid project together, and then Shelly, who helped with the CSF study, and many collaborators that allowed this to happen quickly. STEMCELL Technologies, Kimia and Amanda, for inviting me to give this talk, all the patients that provided different samples for us, and our funding mechanisms to allow for this project to happen. So now I'll take any questions that you guys might have, and thank you guys for your time.
Thank you very much, Eric, for a really great talk. So this concludes the presentation segment of the webinar. And we're now going to begin the Q&A session by addressing some of the questions that were submitted throughout the presentation. And there were quite a few, which is great. So we'll try and get through all of them. And if there's any that we don't get through, Eric will be answering those in written format after the presentation, and we'll send you the answers to those questions once we have the recording available. So as a reminder, if you do have any questions, you can use the chat box on the right of your screen, and we'll pass those on to Eric to be answered as well. So let's start with our first question. So obviously there have been lots of reports of COVID-19 patients presenting with a myriad of symptoms across many different systems in the body. And the first question we have is, are you aware of any reports of symptoms such as dizziness or vertigo in COVID-19 patients?
Yeah, so I think if we look at the literature, there are reports of like dizziness and vertigo that are associated. It's still unclear whether that's due to direct neuroinvasion or possibly due to, you know, like a secondary symptom from like cytokine storm or even like hypoxia, right? I think there still needs to be a lot more kind of studies done, and I think that there is a lot more important like clinical studies that need to be done to be able to characterize it in more detail. And yeah, I mean, that's as much as I know about that also. So there still needs to be a lot more work done.
So you mentioned hypoxia. The second question here is, has there been any evidence yet of hypoxia-associated neuronal cell death in COVID-19 patients, or is that still to be determined?
Right, so there's some pathologic studies where they've looked at post-mortem brain tissue sections, and they've seen like kind of localized hypoxia with microbleeds associated with it. Once again, people don't really know if this is due to some sort of, right, because there's a lot of other diseases with like clotting disorders and stuff that's associated, that if it's due to like mini strokes or actual neuroinvasion within in the central nervous system. But there are definitely localized hypoxia that people see. And the other difficulty in studying this really is that to be able to study that, you need kind of post-mortem samples, right? And there's no kind of like regulated standards of like when the samples are collected and stuff like that. So it's really hard to kind of get to the detail disease mechanism in patients at least, which is why the organoid and stuff like that is very helpful for modeling it.
So on that line, to what degree can those brain organoids that you use in your study mimic the neuroinvasion through the blood-brain barrier?
Right. So our organoid model doesn't have any endothelial cell compartment in large numbers, or the actual blood-brain barrier that's present. So we can't model that very detailed, which is a large limitation to studying SARS-CoV-2 neuroinvasion. There are kind of like ongoing theories on different routes that SARS-CoV-2 can get into the brain. One is through the blood-brain barrier, like you mentioned. The other might be through, you know, the nasal passage, right? Because a lot of people are seeing loss of olfactory sensors and stuff like that. And it can also go through possibly kind of like the sympathetic nervous system that's connected from like the brain to the lung. That's another field that needs to be really kind of like studied and figured out. But yes, it is a limitation to our organoid system. But we were really focused on kind of understanding what happens to the neurons after infection, which is kind of a good system to study that.
And so there's two follow-up questions here that are related. So the first is, have you done any mapping studies then to investigate the location of infected neurons within the brain and which part of the brain is mainly affected, if you have?
Right. So we haven't done a careful study yet. At least in the mouse, I'm assuming this is about the mouse models that we've used.
I think so, yeah.
In studying the mouse models, we do see pretty much every region of the brain affected. However, this is kind of like it's artificial in a way, because the ACE2 expression, the receptor expression, is not the endogenous expression, right? So this will have to be done with more like mouse adaptive viruses in the future, or other techniques, in order to kind of recreate the endogenous ACE2 expression. But the other side to this is that ACE2 expression in neurons and different cell types in the central nervous system is very debated, and which cells are expressing it in mice versus humans is also slightly different. So that raises a lot of questions on how we can best model it, and it'll need to be done in more detail too.
Absolutely. So the other question here that relates to the organoid model that you were using is, were you able to determine exactly which cells in the organoids were infected?
Yeah, so we were able to map the transcript reads of the SARS-CoV-2 onto our single-cell RNA-seq data, and saw actually a very wide range of cells getting infected. It didn't seem to have strong tropism towards one cell type or the other. This is kind of like, I guess, like controversial too. There's different reports on which cell types are getting infected. At least on the transcript level, we're seeing a very wide range, and it's supported by our staining afterwards. But we still don't know exactly like specific cell types and which one it might have more tropism towards.
Okay. This one, it might be a little bit too early to know the answers to yet, but has there been any evidence of SARS-CoV-2 persistence in the brain or the peripheral nervous system?
Right. I think persistence is, yeah, a little too early. Especially there's minimal work showing ability of SARS-CoV-2 to actually neuroinvade, so that's still very debated, and that's something also we're working on. But there are a lot of kind of cured or like patients who've recovered patients with persistent symptoms that have a lot of overlap with different neuropathies that can be present. So it'll be an interesting thing to kind of continue observing and looking at. I think if SARS-CoV-2 can actually invade the central nervous system or even the peripheral nervous system and do significant damage, you would expect more of these cases to be kind of widely observed throughout as we kind of get past this pandemic.
Is SARS-CoV-2 infection in neurons dependent on TMPRSS2?
Right. We didn't test that, so we don't know the exact answer to that. But it would be something interesting to look at in the future.
Okay, so a couple of questions related to antibodies. So the first is sort of a technical question. What antibodies did you use to detect SARS-CoV-2 in your studies?
So most of our studies are done with the nucleocapsid antibody from GeneTex. There are other antibodies that work also, but we just use that one because that one's been available and works the best in our hands.
Okay. And we have a question here from a pathologist in the audience. So she said that in their postmortem cases that they've looked at, so 22 cases so far, they found patients to be SARS-CoV-2 positive using PCR, but using different antibodies for IHC, all cases were negative. And in contrast to that, all the lung samples that they saw were positive through both PCR and IHC. And she's wondering what your opinion is on this discrepancy or perceived discrepancy.
Yeah, I think it's really hard to say. And I would love to talk to her more offline about kind of their findings and kind of talk about our efforts on doing a similar study. But basically, I think it's like you said. I don't think this neuroinvasion is kind of happening in all patients, and to what degree and to what level it's happening, I think it's also hard to kind of figure out. And it goes along with the persistence, right? If it's happening acutely or if it's already cleared or how long it's lasting for. And I know like imaging is always really hard to kind of identify stuff because if it's not prevalent, like very hugely prevalent everywhere, you're sampling a very small section, right, to try to figure out if it's actually there or not. So I feel like there might be kind of a limitation to sampling that might be occurring. I don't have any like opinions on like technical aspects or anything, but it's definitely something I'd be happy to discuss more
Great.
if you email me or something after.
Yeah, we can definitely set that up. So similarly, along the antibody frame of questioning, have you tried studying antibody formation in the patients at different time periods, as there have been reports of diminishing antibodies in COVID patients?
Right. So for our studies, we did not look at like a temporal relationship of the antibodies. That is an ongoing study that people in our lab are doing and other labs are doing, but not in our patients.
Okay. And then there's quite a few questions here about some of your CSF data. So the first one is, for your CSF data, what can you tell us about the disease severity of your COVID-19 patients?
Right. So they were varying degree of symptoms. So some just even had as simple as just like a headache or a little bit of just other neurological symptoms, but none of them were, I would say, like full-on encephalitic. I think one patient was in the ICU eventually. One patient passed away, but there isn't like an underlying theme in the severity or the symptoms associated with our patients.
And did the CSF from the COVID-19 patients that you looked at contain viral RNA?
No. So none of the patients that we looked at had viral RNA. And I'm actually not really aware of too many. I don't even know if there was any studies showing viral RNA of SARS-CoV-2 in the CSF of patients, from what I know.
Okay. So how do the inherent immune or in general cellular makeup of PBMCs differ from the CSF? And is it surprising to you to see different responses between the two sample types? And do you think this is specific to COVID infection or likely possible for other viruses as well?
Right, that's a great question. I think, right, so there would be definite inherent differences between the two compartments, right? And how they behave would be very different in our imagination. So I guess the thing that was really surprising for us was that in a single patient, right, when you have like an infection or something, you kind of imagine that you're getting an overall immune response that's kind of fighting the infection. But what our data is pointing to, and we have some mouse data to actually support this also, that I didn't show today, is that even within a single organism or a single person, you can have kind of like micro immune responses that are occurring that are individual in the central nervous system and another part of the body. And I think that's very fascinating because thinking about kind of in the future, when we're thinking about treating diseases that are more systemic or multi-compartmentalized, you should really consider treating every portion differently or strategically instead of kind of thinking about, oh, we need to treat the patient's viral infection. So I think
So I think that,
that's what, yeah.
yeah. So that leads us to, I think, what'll be our last question, and everything else we'll address in a different format. So the last question is, how does your research overall inform the way that we think about the long-term consequences of COVID-19 and the care that patients need, which is something that you just referred to?
Right. So I think thinking about this, thinking about the patient holistically is very important, and not just kind of thinking about the traditional sense of what a respiratory infection might lead to, but start thinking about how individual compartment in the body can be affected, and then thinking about what the long-term consequence and how we can treat each symptom individually is very important. And I think our studies kind of point to at least thinking about the central nervous system as a key player. And especially because the central nervous system is like a master regulator of many organ functions, right? So I think it's important to consider how it might affect some of the other secondary organ functions if you do have some sort of damage in the central nervous system.
Okay. All right, well, I think that's all that we have time for today. I want to thank everyone for attending, and I'd especially like to thank you, Eric, obviously, for the time that you took with us today, and a really great and informative talk on obviously a field that is moving very quickly. So it's great to have these sort of impromptu conversations. Thanks again, everyone. Goodbye.
All right, thanks, Amanda. Thanks, Kimia. Bye.